Shri Gurubhyo Namaha children, I am happy to welcome you all for our online teaching academy Ekalaiva. For the class 12, subject Biobotany, chapter 4, Principles and Process of Biotechnology. Children, today we are going to discuss about screening for recombinants. You all know about recombinant. What is recombinant? Where you heard the word recombinant? Recombinant is nothing but a bacterial cell having vector. Okay, you just separate this, isolate this vector and insert a gene of interest in that plasmid vector. Now we are getting the recombinant DNA or recombinant plasmid or recombinant vector, right. Now we are going to discuss about the screening for recombinant, whether the recombinant vector is present inside the host cell or not. Clear? So after introduction of rDNA is nothing but recombinant DNA. DNA is nothing but recombinant plasmid. Okay. So after introduction of rDNA into the host cell it may be a plant cell or any bacterial cell. Okay. So after introduction of rDNA into the host cell, it is very essential to identify, very important one. We have to identify those cells which contains that particular rDNA or not. So we have to identify those cells which have received rDNA molecule. This process is called screening. Clear children? Then the vector or foreign DNA present in recombinant cell express the character, while the non-recombinant cells do not express the character or traits. For this, some of the methods are used for the screening. Here, the first one is blue-white selection method. So blue-white selection method is a method used for the screening for recombinant. Shall we move? The first method for the screening of recombinants, that is insertional inactivation. You know the meaning of activation. Here inactivation by adding XII enzyme. Now the particular vector or rDNA is inactivated. So insertional inactivation or blue-white colony selection method. This is the first method for screening for recombinants. Okay children? See, this is the first and very important and powerful method used for screening of recombinants. In this method, a reporter gene, that is XII is a reporter gene, is inserted in the vector. You know very well, vector is a transporting material, for example plasmid. So the reporter gene XII is inserted in the vector. Okay, what will happen? So the vector containing XII is enter into the host cell. What is the main role of this XII enzyme, the XII gene? This XII reporter gene encodes the enzyme beta-galactosidase. So wherever the word ends with ASE means enzyme. So this XII gene encodes the enzyme beta-galactosidase and contains several recognition sites for restriction enzymes. So this gene, XII gene encodes the enzyme beta-galactosidase. Clear children? And this enzyme, what enzyme? Beta galactosidase breaks the synthetic substrate into an insoluble blue color product. Once again I am repeating, the gene LAC-Z gene encodes the enzyme beta galactosidase. This beta galactosidase breaks the synthetic substrate. Breaks means it divides, it splits into the substrate called XGAL. So, XGAL is the substrate. It is a combination of 5-bromo 4-chloro indolyl beta D galactopyridinase. In short, it is called as XGAL. So, this enzyme, what enzyme? Beta galactosidase breaks the XGAL into insoluble blue colored product. Very, very important. The main function of this enzyme is, this enzyme breaks the XGAL into insoluble blue colored product. Okay. So, this enzyme is synthesized by the gene called LAC-Z gene. So, this particular gene encodes the enzyme beta galactosidase, and this beta galactosidase breaks the synthetic substrate XGAL into insoluble blue colored product. Can you get my point, children? Shall we move on to next slide? Yes. See the picture here, children, and this is LAC-Z gene. This is a functional gene. This particular gene encodes the enzyme beta galactosidase enzyme. This is a functional enzyme. This enzyme acts on this substrate. It converts into insoluble blue colored product. So, this beta galactosidase enzyme breaks the synthetic substrate. This is a synthetic substrate. Can you get my point, children? So, this enzyme act on this XGAL. XGAL is nothing but synthetic substrate. It breaks the substrate into insoluble blue colored product. See here. See another picture here, children, and this is a LAC-Z gene. By inserting some foreign gene, now the gene is not in function. It cannot able to produce enzyme. What enzyme? So, there is no enzyme at all. Beta galactosidase. This enzyme is not at all able to produce by this gene. So, there is no enzyme, there is no formation of XGAL into product. Now, it is look like a white colony, right, children? So, therefore, no blue color will develop because beta galactosidase is not synthesized due to the inactivation of LAC-Z. How the LAC-Z is inactivated? By inserting any foreign gene, by inserting foreign DNA. Now, the LAC-Z is inactivated, it cannot able to synthesize beta galactosidase enzyme. So, if there is no beta galactosidase enzyme means there is no breakdown of synthetic substrate into product. Clear children? See, therefore, host cell containing rDNA form white colored colonies on the medium contain XGAL. If the medium containing XGAL, there is no enzyme means it will produce white color colonies, whereas the other cells containing non-recombinant DNA will develop blue colored colonies. On the basis of color colony, the colony color, the recombinants can be selected. This is one important method, screening for recombinant. Okay. See the picture here, this is a plasmid vector, you know very well, by inserting foreign DNA. See here, insert within the LAC-Z. So, this is the LAC-Z gene, foreign DNA is inserted within the LAC-Z. Here foreign DNA is inserted outside the LAC-Z. There is no insertion of foreign DNA. If there is no insertion of foreign DNA means the LAC-Z is functional. It can able to produce a functional enzyme beta galactosidase, and this can able to give blue color colonies. Okay. So, this XGAL on the action of the enzyme beta galactosidase breaks and converts into, breaks the substrate, this enzyme breaks the substrate XGAL into insoluble blue color colonies. Clear? Here second one, insertion outside the LAC-Z. Now the LAC-Z is also active, okay. It can able to synthesize enzyme, and this enzyme breaks the XGAL substrate into insoluble blue colored colonies. Here this method here, insertion within the LAC-Z. So, the LAC-Z is arrested, it is inactivated due to the insertion of foreign DNA, very important. Due to the insertion of foreign DNA within the LAC-Z, now the LAC-Z is inactivated, it cannot produce any enzyme. So, XGAL, the substrate will not converts or breaks into blue color colonies, as it is, it gives white colonies. Culture with the XGAL gives white colonies. Clear children? Second method, replica plating technique. Replica, what is the meaning? Have you heard the word replication? What is replication? Where you heard the word replication? It is one of the main mechanism in DNA, right? Replication, transcription, translation, that is central dogma of protein or cell. So, replication is nothing but a DNA replicate itself to form DNA, right? So, a double stranded DNA is converted into another double stranded DNA, daughter DNA, exact carbon copy. Double stranded DNA give rise to double stranded DNA, the exact carbon copy, that is called replication. Yes, the same here. Replica plating, replica. See here, a technique in which the pattern of colonies growing on a culture plate is copied. Okay, that is called a replica plating technique. A sterile filter plate, the filter plate is pressed against culture plate. Culture plate means that plate containing culture medium, the bacteria are growing. Okay. So, a sterile filter plate is pressed against the culture plate and then lifted. Then the filter is pressed against a second sterile culture plate. What will happen? It will once it take from the first plate and the filter plate is pressed against the second sterile culture plate. The results in the new plate being infected with cells in the same relative positions as the colonies in the original plate. See the picture here, children. This is the master plate, plate 1. Okay, and then sterile filter plate is pressed here, and then that sterilized velvet surface pressed on the master plate and then pressed on the plate 2, second plate. What will happen? See here, here the velvet is free. Once we pressed on the first plate, all it is or goes here, again this pressed here comes here only. So, the replica plate minimal medium having incubation period. So, see the replica plate here, missing on replica plate nutritional mutant takes place. Here the incubation of master plate present on complete medium. Here some missing is there in the second one, replica plate 2. Okay. Usually the medium used in the second plate will be differ as that of the different from the first one, first plate. Two different plate we are using now. In the first plate we are using some other medium, and second plate using some other medium. Okay. So, it may include an antibiotic or without a growth factor. In this way, the transformed cells can be selected. See the picture here, the sterile velvet on the block. This is a master plate, first plate. Pressed over here. Now it is replicate on fresh media, then replicate on fresh media, it is lacks ampicillin, it contains ampicillin, incubate this, then ampicillin sensitive colonies are seen here. Okay. This is called a replica plating technique. This is a second method for screening for recombinant. Clear, children? The next one is molecular technique, isolation of genetic material and gel electrophoresis. Children, what is genetic material? Genetic material is nothing but DNA, deoxyribonucleic acid, right? So, isolation of DNA and gel electrophoresis with the help of agarose gel. Electrophoresis means with the help of electric current, electricity. So, isolation of DNA with the help of current, with the help of electricity, that is called gel electrophoresis. Okay. So, electrophoresis is a separating technique, isolation and separation technique. So, electrophoresis is a separating technique used to separate different biomolecules. Different biomolecules means carbohydrate, proteins, DNA, RNA, like that. So, it is used to separate different biomolecules with positive and negative charges. Clear? So, DNA is usually negative charge. Clear? Principle. By applying electricity, DC, that means direct current, the molecules migrate according to the type of charges they have. So, just we apply the current, electricity, the molecules are moving, they are migrating according to their charge. If it is a cathode means, cation means moves towards the cathode, anion means it moves towards the anode, like that. Clear? So, by applying electricity, that is DC, the molecules migrate according to the type of charges they have. The electric charges on different molecules are variable. So, RNA have different charge, protein have different charge, DNA have different charge. So, each one biomolecules have different charges. So, in that first one, agarose gel electrophoresis, here the gel, agarose gel you are using, that is so called as agarose gel electrophoresis. It is mainly used for the purification of specific DNA fragments. So, if we want to purify the specific DNA fragments, we just go for agarose gel electrophoresis. Agarose is a convenient gel for the separation of DNA fragments ranging from size a few hundreds to 20,000 base pairs. So, agarose is a convenient gel used for the separation of DNA fragments size from a few hundred to 20,000 base pairs. Another gel is there, polyacrylamide gel. Polyacrylamide PEG is preferred for the purification of smaller DNA fragments. The gel is complex network of polymeric molecules. A DNA molecule is negatively charged. Okay. So, DNA is usually negatively charged molecule. Under an electric current, electric field, DNA molecules migrate through the gel. Okay. The advantages of agarose gel electrophoresis are that the DNA bands can be readily detected at high sensitivity. See the picture here, and this is the apparatus used for agarose gel electrophoresis. The main advantage is we can able to detect the DNA bands with a high sensitivity, and the bands of DNA bands or bands of DNA in the gel are stained with the dye, very important one more question, ethidium bromide. Just using this dye we can able to stain. The bands of DNA in the gel are stained with by using the dye called ethidium bromide, and the DNA can be detected by a visible fluorescence illumination in UV light will grow orange fluorescence, which can be photographed. Okay. With the help of this, we can able to separate the DNA fragments or isolate the DNA fragments. Thank you children. We will see in the next class.